Hello, my name is Brian Ross. I am the head of biochemistry at the Quantum Biology Institute, and I'm very excited to share with you our work on searching for magnetosensitive proteins to take quantum measurements in live biological systems. Biological systems can sense weak magnetic fields. These are fields on the order of tens of microtesla all the way up to tens of millitesla. So the magnetic field of the Earth is 50 microtesla. The magnetic field from your iPhone is in the tens of millitesla. And it's very interesting that biological systems can sense those magnetic fields and respond to them. And there's been particular interest in studying organisms that can do magnetoreception, that they can sense the magnetic field of the Earth and use that information to help them navigate. We know that migratory birds, insects, sea turtles, monarch butterflies, for instance, can do this. And there's been a lot of interest in the field for studying magnetoreception. But it's not limited to magnetoreception. Lots of other biological processes are dependent on weak magnetic fields and change when you change the magnetic field conditions. This includes embryo development, redox biology and metabolism, cell cycle regulation, circadian rhythms, DNA repair, and many, many other processes. There are three potential mechanisms that have been demonstrated to explain how biological systems could detect a magnetic field as weak as that of the Earth's magnetic field, around 50 microtesla. The first is electrical induction. So organisms can move throughout the magnetic field, and the changing magnetic field as they move through it induces an electric field which they can detect. And a good example of that is sharks. They have small little organs called ampullae of Lorenzini. And as they move through the water, they move through the magnetic field of the Earth. They induce electrical fields, which they can then detect. There's also magnetite, which is crystals of iron oxide. They can act as little bar magnets and they can align with the magnetic field of the Earth. And we see that in particular types of what are called magnetotactic bacteria. These magnetite crystals have to be large enough to overcome thermal movement, and they're found in bacteria, but they don't explain magnetosensing in other types of organisms. The third mechanism, and that's the mechanism that we're primarily interested in, is the radical pair mechanism. And this is fundamentally a quantum mechanism based on electron spin. So essentially, light can kick off a process by which an electron donor donates an electron to an electron receptor that creates a pair of radicals. And that pair of radicals can exist in different spin states, and external magnetic fields can change what's called the superposition between these different spin states, and that leads to different biological outputs. The evidence that we have for magnetic field effects in biology come from two very different length scales. So we have physiological information from animals and plates of cells demonstrating the phenomena. So these are gross endpoint experiments. And we also have molecular information on the order of nanometers. This is in terms of magnetosensitive proteins and understanding the electron spins. The problem is that we don't have a lot of information connecting that gap. How do we get from magnetosensitive proteins to the gross physiological changes that we see in the cell? What pathways, processes are involved to link magnetosensitive proteins to changes in physiology? For the quantum mechanism for detecting weak magnetic fields, we face objections from two different scientific communities. Biologists will point out that weak magnetic fields may be too weak to have a biological effect. They may point out that endpoint studies on magnetic field effects in organisms may not be perfectly controlled. And so we're not sure these effects are truly due to the magnetic fields or they're due to other compounding variables that are difficult to control. Physicists, on the other hand, may argue that coherent superpositions that are needed to carry out this mechanism require low temperatures and isolated systems. And biological systems are the opposite of that. They're warm, wet, they're busy, and they may not allow for a coherent superposition to be maintained long enough to have an effect. To address these concerns, our solution is to take quantum measurements of spin superpositions in live cells under physiological conditions using custom quantum instruments. To support QBI's research into the radical pair mechanism, we have our biological research program, which aims to find the biological samples that will be best to study using the custom instruments that we're developing. And we have 3 different paths of our biological research program. We are interested in finding magnetosensitive proteins. So we're asking which proteins respond to weak magnetic fields and why. We're doing an omics approach. This is looking at the top down and trying to understand which biological processes are affected by weak magnetic fields. And lastly, we have a research program on redox and metabolism. So we want to understand the role of what are called reactive oxygen species. These are oxygen byproducts of metabolism and other cellular processes. And we'd like to know what their role is in sensing weak magnetic fields. As part of our mission at the Quantum Biology Institute, we publish our research as we go, including intermediate results. And we do this through micropublications. And we submitted our first micropublication to bioRxiv. And so it's available to the public. And here's a link here. There's a link here if you would like to access it and read it. So we're particularly interested in a family of proteins called flavoproteins. These are proteins that bind molecules called flavins. And flavins are derivatives of riboflavin, which you may be familiar with as vitamin B2. But in cells, these molecules function in 2 different forms. One is FMN, or the other is FAD. And these are cofactors found in proteins across the tree of life, and they're involved in many different biological processes. And what's interesting about flavins is that they can be reduced by a single electron to form a radical, or they can be reduced by 2 electrons to form a fully reduced state. And because they support stable radical form, they can participate in the radical pair mechanism. And there's a diagram here on the right demonstrating the radical pair mechanism. You have a flavin that with an electron in the ground state, and blue light excites that electron and brings it to an excited state. When it's in the excited state, it can then relax back down to the ground state, and that gives off light in the form of green fluorescence. Or you can have electron transfer from a donor molecule, for example, tryptophan, but it could be another electron donor, to the flavin. So when you have electron transfer from the tryptophan to the flavin, you get what's called a radical pair. So there's a radical from the tryptophan losing an electron and a radical from the flavin gaining an electron. These radicals are what are called spin correlated. So their spin exists in a coherent superposition between different states. They can be in a singlet state where the spins cancel one another, or they can be in a triplet state where the spins add together. And what's interesting is an external magnetic field can modulate the superposition between the singlet and triplet states. And because singlet and triplet states lead to different chemical processes and ultimately different biological effects, that allows the magnetic field itself to be able to have a biological effect. What's also interesting is that the singlet population can more easily decay back to the ground state, which then can feed back in to the process of fluorescence. So in this way, our fluorescence intensity is a proxy readout for the singlet character. So in essence, using fluorescence measurements, we can get information about the spin states of this radical pair. The magnetic field effects have been studied in a handful of different proteins. The primary protein that's of interest in this field is called cryptochrome, and that's a protein that's involved in circadian rhythms. And it's of particular interest because it is expressed in the retina of certain types of migratory birds. And it is the best supported hypothesis for explaining how these birds detect the magnetic field of the Earth and use that information to help them navigate. An evolutionarily related protein to cryptochrome is called photolyase, and it's also been shown to have a magnetic field effect. But photolyase, instead of being a circadian rhythm controlling protein, is actually a DNA repair enzyme. So photolyase can mediate magnetic field dependent DNA repair. Certain optogenetic tools like love domains have also been shown to be magnetosensitive. And through engineering, it's been shown that you can actually amplify the magnetosensitivity. And lastly, fluorescent proteins such as M scarlet 3 and other fluorescent proteins, which are not flavoproteins in and of themselves, have also been shown to have magnetic field effects when in the presence of exogenous flavin, which is very interesting because these fluorescent proteins are used widely throughout cell biology research. So we aim to take quantum measurements of spin photophysics and connect that information to biological outcomes. And to do this, we need to find candidate proteins for taking quantum measurements using our custom instruments. These proteins should have a strong fluorescent signal that's sensitive to magnetic fields due to the radical pair mechanism. And ideally, they would mediate biological processes that have a clear physiological effect on cells so that we can connect spin physics of particular electrons on a particular protein to physiological effects in the cell. We also aim to understand the molecular basis of magnetosensitivity in proteins. So we'd like to understand what molecular characteristics determine their magnetosensitivity. How can we modulate their magnetosensitivity through mutation or through changing the environment of the protein? And how can we exploit this knowledge for technological development and human use? We aim, finally, to understand how common this phenomenon is in the biological world. Is this unique to the handful of proteins that have been studied? Or this is a much wider phenomenon than previously believed? So we'd like to understand what biological processes are mediated by magnetosensitive proteins, what families of proteins, in addition to flavoproteins, have the potential to be magnetosensitive, and how can we predict magnetosensitivity based on structural or sequence characteristics of the proteins. I'm going to focus now on our work characterizing a protein called maglev2 and trying to determine whether or not maglev2 is a good candidate for doing the quantum measurements in biological systems. So maglev is an engineered magnetosensitive protein. It was developed by collaborators of ours in Northern California, and it was developed from ASLEV2, a photoreceptor in plants. So ASLEV2, it's part of a phototropin, which is a protein that plants use to detect light and grow towards the light. And this particular protein, ASLEV2, was engineered through rounds of mutation and screening to isolate variants with large fluorescent changes to magnetic fields. In this engineering process, the native function of the protein was lost, but what came out of it is a fluorescent protein with large changes in fluorescence due to low magnetic fields. So we aim to fully characterize the fluorescence intensity as a function of magnetic field strength in maglev2 and to see whether or not that function is consistent with known theoretical models of the radical pair mechanism. So in our work, we took a plasmid, a circular piece of DNA, with the maglev2 gene, and we transformed E. coli with that plasmid and induced them to express the maglev2 protein. We plated this bacteria on bacterial plates and then imaged the proteins using a custom bacterial plate fluorescence imaging system that we're calling the Bacterioscope. We then did colony detection and particle analysis to extract fluorescence traces from the bacterial colonies. And we get then a curve that looks like this. So the black curve is the fluorescence trace. The teal bars reflect when we apply an external magnetic field. And you can see when we apply the external magnetic field that there's an acute change in fluorescence intensity as a result of the external magnetic field. But you can also see that there's an overall decrease in fluorescence as the experiment goes, and that's due to photobleaching and reversible photophysics. And so we want to isolate just the change that's due to the magnetic fields in and of itself. So we did curve fitting of the off segments, and then we subtracted out the baseline, and we're able to isolate just the changes in fluorescence that are specifically due to the magnetic fields that we applied. So when we did these experiments, we saw something interesting. At very low fields, such as at 1 millitesla, you actually see an increase in fluorescence when you apply the magnetic field, which then reverses when we remove the magnetic field. So at very low fields, you have a positive effect. But this is data taken from the exact same colony at 2.5 millitesla. So data taken from the exact same colony at 2.5 millitesla had the reverse effect, where you see a decrease in fluorescence when you apply the magnetic field, which is then reversed when we remove the external magnetic field. So you can see when we plot the magnetic field strength to the magnetic field effect is that you see a change in sign. At very low fields, you have a positive effect, and then it changes sign to a negative effect. So we thought that was very interesting. So we decided to fully characterize the relationship between the magnetic field strength and the fluorescence intensity. And we did that through ramping experiments, where we ramped the magnetic field intensities from 0.5 millitesla all the way up to 100 millitesla. And we compiled the data from several of these experiments to get a curve that looks like this. Without the maglev2 gene, we don't see any effect of magnetic fields on fluorescence intensity. However, we see a very particular curve when we have the maglev2 protein there. At very low fields, if we zoom in at the very low fields, we see the kind of pattern that we saw before. At very low magnetic field strengths, you see an increase in fluorescence as we increase the magnetic field strength. It reaches a maximum, changes direction, and starts to decrease. At around 1.5 millitesla, the sign changes from positive to negative. And above 1.5 millitesla, you have a negative magnetic field effect, or a decrease in fluorescence due to applying an external magnetic field. What's also interesting is that the effect plateaus at around 60 millitesla. It's around the same magnetic field strength as that of your cell phone. So what's interesting about that is that it means that this effect is truly an effect of weak fields. Applying stronger magnetic fields does not result in a stronger effect. And this, what's called non-monotonic behavior, the behavior of it increasing, then decreasing and changing sign, the fact that it's a low field effect, that it doesn't get stronger with stronger magnetic fields, is fully consistent with theoretical models of the quantum radical pair mechanism. So our conclusion is that maglev2 is a good candidate for taking quantum measurements of spin superpositions in live cells. It has a very large fluorescence signal, and that fluorescence is strongly magnetosensitive with changes of up to 20%. The response is consistent with radical pairs, specifically radical pairs born in the triplet state. So we learned something particular about the photophysics from this experiment. So as a result, we conclude that it's a good starting point for developing the methods and calibrating the instruments that are used for taking quantum measurements in biological systems in the warm, wet, complex environment of the cell. However, maglev2 is an engineered protein that does not preserve its biological function. So this motivates a search for a natural magnetosensitive protein involved in biological processes that we may want to investigate using our instruments. So our next steps are to further characterize the maglev2 protein, and in particular, its magnetosensitivity. We'd like to measure the fluorescence intensity versus magnetic field relationship, not only for those magnetic field strengths above the magnetic field of the Earth, like we did in this micropub, but we'd also like to go into hypomagnetic conditions, those magnetic field strengths below the magnetic field of the Earth. And we hypothesize that the trend will continue. We'd also like to test how mutations and the environment affect the magnetosensitivity and in general affect this curve, so that we can understand how we can modulate the different parameters of maglev-2's magnetosensitivity. And in doing so, we may learn how we can predict what other proteins may be magnetosensitive. Once we've done that, we'd like to perform quantum measurements in maglev-2. So that's taking nanosecond resolved single photon measurements of the fluorescence of maglev-2 and try to measure at the nanosecond timescale these quantum superpositions in biological environments. Lastly, we'd like to test our hypothesis that magnetosensitive proteins are widespread in biological systems, that it's not just unique to this handful of proteins that have been studied, but that this is a much broader phenomenon in biological systems. So to do that, our plan is to screen all flavoproteins within one particular species. We'll start with E. coli, and to screen other photoreceptors for magnetic field effects, to see if we can find a natural magnetosensitive protein that could be good for taking such measurements. Anyway, thank you very much for joining and for learning more about the quantum biology in general and about our work with magnetosensitive proteins in particular. And I look forward to interacting with you and answering any questions you may have. And thank you very much.